हेलो हेलो। चल रहा है ना? लाइव। हाँ। स्टैंडर्ड टाइम इज 5 मिनट्स ऑफ। बाकी ऑन नहीं है, ठीक है। 2 मिनट 2 मिनट। माइक। শুনে একটা ভালো দেখে একটা মেসেজ করে দেবে। Okay, one of our most regular members in, so I can start. But that's the assembly time. So, okay. So, thank you all for coming for this week's Wednesday colloquium. And I am very pleased to have a very good friend, Professor Shantanu Mukherjee from Indian Institute of Science, Bangalore, visiting us today for this colloquium. So, for those who are attending the colloquium for the first time, either through YouTube or Zoom or in this room, including Shantanu. The idea of this Wednesday colloquium is pretty old, and it was started by Professor Homi Bhabha, our founding director. The idea was to bring all the people together, the natural sciences faculty members, for one day, and Bhabha could at least meet over lunch, everybody, just like the lunch you had, and meet all of some of the members, and also attend a colloquium in any of the disciplines of physics, chemistry, and biology. At that time, there was no distinction of chemistry such, but it was called chemical physics at that time. But over the years, this has been an age-old tradition, and I'm thankful you're contributing to this. And before Shantanu delivers the talk, I would like to briefly introduce Shantanu and to the audience here. So Shantanu actually did his bachelor's in chemistry with honors from Ramakrishna Mission Narendrapur, and this is part of University of Calcutta that gives the degree. And he did that in 2000 actually. Um, then he went to do his master's from IIT Kanpur Department of Chemistry, which he finished in 2002. And after that point on, he went to do his PhD from University of Cologne. And this is the place where he sort of started doing organic chemistry in all the full glory. And this is with Professor Albrecht Berkesel. And Shantanu did some wonderful work with Professor Berkesel. He was summa cum laude in the diploma, the degree that he got, one of the top students from that place. And then his journey from Cologne took him to Max Planck, where he worked with Benjamin List. And if some of you may recollect, Ben List won the Nobel Prize a couple of years back for his fantastic work on organocatalysis. And when Shantanu had joined, he was still a young person, and they were discovering new ideas with how to use chirality, how to induce chirality in organic reactions. So working with Ben List, he spent like 2 to 3 years 2 years. And then he moved to the lab of Elias J. Corey, E.J. Corey, who is very famous amongst some of our chemists, because you have heard about Corey in textbooks. And E.J. Corey had been one of the most pillars of organic chemistry in the last century. And he and Woodward were one of the famous couple who would take organic chemistry to new heights, both from the point of view of understanding of why organic chemistry works this way, as well as developing new methodology and reaction tools. Well, getting trained from these luminaries in the field, Shantanu started his own lab in 2010 in IISc Bangalore. And he is there as a professor now ever since for last 14 years. Now he has completed 14 years there. And Shantanu has set up a research program that is world renowned now for building chiral frameworks, and this is as well as reaction methodologies while doing that. And he has of course been rewarded with many awards, but I would just like to mention two of them. One is Shantanu is excellent teacher. You will see that. He has won the teaching prize in IISc Bangalore, which is, I think, one of the reputed prizes that one can win in 2014. And also, he has been sort of the associate editor of Organic and Biomolecular Chemistry. He has been associated with now, of course, SYNLET, as well as Journal of Organic Chemistry in the editorial advisory board. But without further ado, I would like to welcome Shantanu for the talk on breaking symmetry, reaction development, and application to complex targets.
Thank you, Jyotisman. I'm audible, right? Okay. First of all, it's a great pleasure to be in TIFR. And this is my first visit in this, of course, very famous institute. And I thank Jyotisman for inviting me to this Institute Colloquium, or Wednesday Colloquium, as you sometimes refer to. And I know this is a very mixed audience, and I hope I'll be able to keep you engaged through questions and through my presentation. And I'll tell you, so of course, I'll come to the title a little bit later. So our group work on asymmetric catalysis. This is one field of organic chemistry where you start from a chiral molecule, and through catalysis, you generate chirality in the molecule. Of course, you produce the desired targets in enantiomerically enriched form. And there are many areas in this within asymmetric catalysis that we work on. And here I have listed some of them, but without going into the details, there are two major areas of asymmetric catalysis that we have been engaged in since 2010. One field was, of course, organocatalysis. That was much closer to my heart. We have been working since the beginning. And about 5-6 years ago, we also started working on transition metal catalysis. The objective actually of working in two different areas to get sufficient expertise and to develop reactions on our own, to develop some methods so that we can actually combine them to explore the prospect that remains at the interface of organocatalysis and transition metal catalysis. Because combining two different catalysts, you can actually develop some reactions that are not possible by a single catalyst alone. And I will not go through the details of this part today, but as Jyotisman was mentioning, that we are mostly focusing on discovering new reactions, new transformation that is not known yet. And of course, for any of this discovery that starts with a very solid mechanistic working hypothesis, and that requires, of course, a lot of screening of catalyst and reaction conditions and solvent and so on. And this phase, of course, requires a lot of perseverance. There are frustrations and failures for a long time, and one has to be patient. Of course, in some cases, we get lucky. Things happen pretty quickly. And once we do that, we go to check how general the reaction is. And in this process, actually, in both this phase, we get a lot of unexpected findings, which are often called serendipity. And then, of course, we use the tools of spectroscopy, computational methods, and so on to understand the mechanism of the reaction that is happening. And in overall, the knowledge that we generate in that process goes to the drawing board for discovering more new reactions. And of course, we focus a lot on the applications. Today, I'll talk a lot about applications of what we are doing today. But today I'm going to talk about so-called symmetry breaking or desymmetrization reactions. Now, if you think of around us some symmetrical object or symmetrical building or anything, we get fascinated like that. Like one of the probably the biggest example that comes to mind is the Taj Mahal. If you go there, you will see it is extremely symmetrical. And then we see that such a huge monument people built in such a symmetrical manner, it must have taken a lot of effort. It indeed did. But when it comes to the molecular world, building symmetrical molecules are actually much simpler compared to the unsymmetrical molecule, particularly in a enantiomерically enriched form. I will come to that in a minute. And so therefore, one of the ways to do that is you can create a symmetrical molecule and then go on to break the symmetry. And that will create an asymmetry in the molecule, and therefore the job becomes much easier. So today, in the next 40 minutes or so, I will try to give an overview in this area, how symmetry can be broken to make asymmetric molecules. So what I mean by breaking symmetry, to think about this cartoon representation, these two blue balls, what we have, they are like identical functional group. So in this molecule, this is symmetrical, as you can see, there is a mirror plane, and this center is not a stereo center. But if we convert one of these two functional groups to something else, like I have shown with the cartoon representation, then this center becomes a stereo center. Now, there is, of course, a statistical probability. You can functionalize one of them, either one of them, like you can get either the right one or the left one, and they are, of course, mirror images. And you need to do this in a controlled manner. You don't want to do both, because again, you will end up with asymmetrical molecules. That is not what we want. And this type of compounds are called prochiral compound, because this center is a so-called a prochiral center. It is waiting to become a chiral center. There are, of course, other classes of molecules, because we want to make them only one of them. Like these are called meso compound. These are also symmetrical, but now these two are actually chiral centers. But overall, the molecule is not chiral, because they are so-called internal compensation that makes the molecule a chiral, makes symmetric. But once again, if we convert one of them, then we can actually generate multiple stereo centers in one step, and the whole molecule becomes chiral. This type of compounds are known as enantiomer group selective reaction. So these two groups are called enantiotopic, or simply called desymmetrization reaction. There are a lot of advantages of this method is that you can generate multiple stereo centers in a single step, and some cases a stereo center can be generated quite far from the reaction site, and that is something quite exciting, because generally when you do a reaction at a particular point, particular reaction site, it is very easy to control the stereochemistry at that site, because the reaction is happening there, where there's a catalyst or a reagent that is at the very close to that site. But if the reaction is happening here and we are generating a stereo center quite far from it, that is sometimes difficult to control. Now the question is how far the center can be, and you can still be able to control that. Most importantly, why do we even care about breaking symmetry? So I will try to answer some of these questions in my today's presentation. Now, let me give you a simple example. So of this type of species that I was talking about. Now, you think about this molecule. This is, of course, symmetrical. You have the CH2, N2, the nitromethylene group, on both sides. So therefore, this is a symmetrical molecule or prochiral molecule. We can break the symmetry by stereoselective modification of one of the nitro groups. Then the center becomes chiral, or we can actually functionalize this carbon center. So we chose to do the latter one to make something like this. You can generate two different stereo centers. It's called desymmetrizing allylic alkylation reaction. And we have done that with the use of this type of organic molecule. So this is when I mentioned organocatalysis at the few minutes ago. Organocatalysis is a very well-known field now because of the 2021 Nobel Prize. And this has been active, this field has been active for over the past 2 decades, I would say. And so this refers to basically use of pure organic molecules most of the time as catalyst. In some cases, there can be a metal in the molecule, but metal does not participate in the catalysis. It's mostly as a structural feature of the molecule. But in this case, you can see this is a pure organic molecule that is acting as a catalyst. I will come to this type of molecules a little later because this is just an example. I was trying to give you how to break the symmetry. So you are actually forming the CC bond here. That's the green bond. And we can get the product as a single diastereomer. Like you can control the stereochemistry at both these sides with high stereoselectivity. Now, today's focus is going to be this particular reaction. This is another desymmetrization reaction. I will discuss it in a bit more detail. So if you look at this compound, don't look at the H because it is there on the other side. I've only highlighted one. This is also a symmetrical molecule. There is a plane here, and we are forming this red bond in this process. So this is a very unique reaction because we are doing a CH alkylation reaction without using any metal, enantioselectively, and using nitroalkane as an alkyl source, which is also very unusual. People generally use organometallic compound for forming this type of bond. But we showed for the first time that you can use actually nitroalkane to form this type of molecules. And we are once again using a very similar type of catalyst that I showed in the previous slide. And these are called bifunctional catalyst. Let me give you a little bit about what I mean by bifunctional catalyst. Generally, there are two different functional groups, complementary functional groups in the same molecule. Like in one can be a Lewis or a Bronsted acidic, other can be Lewis or Bronsted base. Like in this particular case, this is a Bronsted acid and this is a Bronsted base. So therefore, they're complementary functional groups. And this concept was coined by IISc Takemoto's group back in 2003, and where they actually added a nucleophile to this electrophile to form this bond. This is called a Michael reaction. And typically, when you do the catalytic transformation, so catalyst basically tampers with the orbitals, the frontier orbitals of the molecules. It either, and so normally in a CC bond formation state, it is the filled orbital interact with an empty orbital. In other words, the HOMO interacts with the LUMO of the electrophile. So catalysis can happen either by raising the HOMO energy or lowering the LUMO energy. The overall objective is to bring the gap between the HOMO and LUMO. And this is what bifunctional catalyst does. It enhances the HOMO and raises the LUMO at the same time. So that's what I was showing here, that in this case, because of the hydrogen bonding, these nitroolefins can become more electrophilic. At the same time, this Bronsted basic nitrogen makes this much more nucleophilic. So that means this LUMO energy is lowered and the HOMO energy is enhanced at the same time. So in our case, so this is what we thought the initial mechanism. That's what our hypothesis was. Or once we found out this is somewhat a serendipitous finding in some other context, which we actually brought into this particular transformation, and we could actually do the CC bond formation. So in this case, so like I was talking about the nitro compound, here these two positions are actually enantiotopic. Now, what we thought initially, this nitro alkane can add here either of these two positions in so-called an enantio position selective manner, because if it adds above or below, you will get two different stereo center. And once this addition happens, this proton is actually alpha to the carbonyl. It is acidic, so therefore it can actually eliminate, and along with the NO2 minus goes out, so you form nitrous acid and an exocyclic double bond, which then isomerizes to the thermodynamically stable endocyclic double bond. That was our initial hypothesis. Now, based on this initial finding, we went on to apply the same concept to a different class of substrate, like a meso compound. This actually solves also another problem. This type of compound cannot be synthesized directly by Diels-Alder reaction in an enantioselective fashion. So we make this one via Diels-Alder and then break the symmetry of the molecule. This is exactly what I was telling earlier, that making an unsymmetrical molecule is much more difficult. So you can build the symmetry and then break the symmetry. So in this particular case, the same same mechanism that what I just talked about, we were hypothesizing. And in this case, for one particular substrate, we found that along with the final product, we also got the intermediate that we have been proposing. We could isolate, characterize completely, and once we treat this with an achiral base without the catalyst, we can get the final product with the same level of selectivity. So that shows that this enantiomer position selective addition is indeed the key step of the reaction, or the rate determining step of the reaction. Unfortunately, in this particular, yes, sure.
Why do you need in the same catalyst two different kind of functional groups? You could have separate two different entities with different properties.
Okay. Yes, that's a good question. So if we look at this molecule here, we have this 2 NH group, right? So this is the Bronsted acidic one, and a tertiary amine group, which is a Bronsted basic one. Now, having them in the same molecule, so because if you think of, without looking at the molecule, that I am taking an acid and a base under the same reaction mixture, of course, they would like to interact with each other. So if you have it as a 2 separate molecule, it is much easier for them to come and interact with each other because there is no rigidity in the molecule. So in this particular cases, because of the molecular rigidity, they cannot interact within the same molecule. So that's one of the advantage. Other possibilities, of course, when the 2 substrates come close to each other, their stereochemical outcome becomes much easier. Because if we have the 2 separate molecules, they are activating each of them, then there is a much more flexibility that so in enantioselective catalysis, it is the phase selectivity that is the most important one. Now, if the 2 molecules are completely separate, 2 substrates, and they are coming, so any of the phase can actually approach each other. But if it is within the same molecule, one of the phase will be inaccessible. So that actually makes the stereo control much easier. It has been actually shown with the control experiment that if we take separately, we get much lower reaction rate and much lower selectivity compared to the intramolecular one. Now, unfortunately, in this particular case, we could never isolate or detect this intermediate. So that basically put a question mark to the mechanism that we were thinking, at least in this particular case. So more specifically, we started asking questions like in this case, what is the rate and enantiomer selectivity determining step? And also, how does the catalyst control remote stereo induction? As I was talking about, in this case, the reaction is happening here, but this is the center that we are controlling. So how it is 2 bonds away from the reaction site, and in the other case, it is actually multiple bonds away from the reaction site, and how the stereochemistry is still controlled. So to understand that, we actually combined a experimental spectroscopic technique that is called natural abundance 13C kinetic isotope effect. And this is a technique that was popularized by Dan Singleton's lab, and density functional theory calculations. And we took the help of Jennifer Hirschy from Binghamton. So we collaborated on this project, and I will not go to the details, but I can tell you in this particular case, we found that it is not the addition, it is the elimination of the nitro that is the enantiomer and the rate determining step of the reaction. And so this gave us some idea about how this reaction is happening. But again, it depends a lot on the substrate, because in the other substrate, we saw that the addition is the enantiomer determining step. So now, what we learned in the overall process that nitro can be a good leaving group, so in both these transformation. So therefore, we are thinking like with this knowledge, we are thinking, can we develop some other reaction where the leaving group ability of the nitro can be explored? And this is the reaction that we developed. So what we did, this is the final product we obtained. Now, a normal retrosynthetic analysis will allow us to cut this bond and lead us to these 2 fragments. Now, if we look at these 2 fragments, this is alpha to the ketone or alpha to beta keto ester. This is actually nucleophilic. or the negatively charged. Similarly, the alpha position of an enone is also negatively charged. So therefore, here we have the two reactive centers with the mismatched polarity. They cannot form bond with each other. Because we have a nitro group at this end in this modified substrate, this center now is electrophilic. So therefore, we have converted the mismatched polarity to matched polarity. Now the CC bond formation can take place, and then we can eliminate nitro group to achieve this otherwise impossible transformation. So this is basically what we have, new reaction we have developed based on the knowledge that we have generated in the whole process. Now coming to the, yes, please.
In the last slide that you have showed, so why are you not getting the Michael type addition here?
This is a Michael type addition. So the Michael addition is happening actually at this position.
Okay, got it, got it.
Now, of course, I believe what you are trying to ask is,
Michael is like 1-4, right? Michael addition is like 1-4 addition.
Yes. So this is also 1-4. So okay, let me clarify. So this is the electrophilic center. This center is also electrophilic.
Yes.
So I think you are thinking, if you think of 1-4, you can think of 1-4 with respect to the ketone.
Okay.
Or 1-4 with respect to the nitro.
Okay, got it.
It's not possible. Now, in this case, since we have this catalyst and that forms hydrogen bonds selectively with the nitro one, this center, the beta to the nitro, is more electrophilic compared to the beta to the ketone.
Okay, okay.
So nitro now plays multiple role. What I mentioned here, polarity reversal, it does the regio control of the conjugate addition, like what you are mentioning, and then of course it goes as a leaving group to allow the bond formation.
Okay, got it, got it. Thank you.
Now, having done that, we thought what can be the application of this method. Yes, please.
Sir, in the previous slide, the catalyst, the organocatalyst you are showing, what is the role of the cyclohexane ring that you are using?
Okay, so normally most of the other catalysts I showed, that is this aryl ring. So this, okay, this can have normally, so if we have this bis trifluoromethyl phenyl group, that case, what has been shown that these octoprotons can also participate in a very weak hydrogen bonding. Okay, but that is not always essential. In fact, not always beneficial. So in this particular case, we have tried that as well, and we found that with the cyclohexyl one, we get better selectivity. But we do not have the full explanation why that happens. But in some cases, this is not uncommon because phenyl ring is flat, whereas cyclohexane ring is kind of puckered. So that therefore it can actually play some role in controlling the selectivity, but we do not have the exact idea how that happens.
So is it specific, totally substrate specific interaction you are considering? When you are like you are mentioning that the orthohydrogen is participating. So based on the substrate you are
yes, that will depends on the substrate. In some cases, in the other case, it has been shown particularly with the urea. So sometimes this CH can actually form hydrogen bond intramolecularly with this oxygen, and that makes the catalyst rigid. So that sometimes helps, not always.
Okay. Thank you.
Yeah, I'm gonna.
In the interest of time, let him speak. You have a quick question or?
Yeah, nucleophility is basically a leaving group. It's an old term. So something that goes along with the electron, it's called nucleofuge, and something just leave the electron behind, they're called electrofuge. So this is an old term, what you call a leaving group. So yeah.
What are the different parts, important parts? How do you design it? So you have one catalyst.
Right. Right.
What is the relevance of the other?
In the catalyst. Yeah. So the Takemoto, so in the other case, what we showed, so what essentially we need is an 1,2-diamine. So this catalyst is an 1,2-diamine. Now, in some cases, for some of the reactions, we have done a systematic analysis because there can be a double bond, which we reduced. And of course, the role of this aryl group I have already mentioned. Now, the rest of them, this actually comes from a natural compound. It's from quinine. So if you start from quinine, in 2 steps, you can make this molecule. One is just we reduce this, we convert the OH to the corresponding NH2 and then couple it. Okay. So, but in some cases, we did check systematically that instead of alkene, if we have the hydrogenated one, it is a better catalyst. Then we thought probably it is a steric. we put another group at this position, and that turned out to be a better catalyst. But that again depends a lot on the reaction. It is not a general trend. But at the same time, when we prepare a catalyst, we check two things. One is, of course, how easy it is to make the catalyst. We don't want to make it very complicated. If the catalyst takes 10 steps to synthesize, nobody's going to make that. So if it is 2 steps from an easily available molecule, we can actually make it easily. All right, so now coming to the application part of that. So that's what we thought. We can actually, I mean, generally when we develop a reaction, we apply to some different substrate. The next question comes, what next? Can we apply it to some real life problem? So that actually brought us to this very interesting class of molecule called ladderin phospholipids. Now, this was first isolated in 2002, and it was an exciting isolation, exciting findings, because if you look at these molecules, they look kind of unusual. I mean, they are unusual and almost thought of impossible molecules, because there are multiple 4-member rings fused to each other, and that's why they're called ladderins, because of the ladder-like architecture. And this was isolated from this bacteria called anaerobic ammonium oxidation bacteria, which is responsible for about 50% of the nitrogen that is produced in the ocean. In fact, this bacteria or this class of bacteria can also be found often in the wastewater treatment plant. And what it does, it actually does this reaction. It converts ammonium nitrite to nitrogen. So it basically reduces nitrite and oxidizes NH4 plus to finally come to nitrogen. And this process is an overall exergonic reaction, but it doesn't happen in the open ocean. It happens in a compartment within the bacteria called anamoxosome. And another interesting part is that, so you can see hydrazine is actually an intermediate in the process, and we all know hydrazine is toxic. So what was thought initially, and these ladderins were isolated from this membrane of this anamoxosome, the one that I have highlighted is green. And in 2018, actually, what they found out that it is not, this membrane does not, so what initially was thought that membrane actually stops the permeation of hydrazine across this, from anamoxosome to riboplasm, and because hydrazine is toxic to the cells. So, but in 2018, the Stevens Boxer and Noah Burns group, they actually did a systematic investigation and showed that this membrane is as permeable to hydrazine as any other lipid membrane. Only thing is that it is less permeable to proton. As a result, you can see here, inside the anamoxosome, the pH is 6.3, outside is 7.3. So overall, it actually creates a proton motive force across the membrane and drives the ATP synthesis. And so from this membrane, which is a bilayer, this type of ladderin phospholipids were isolated. And these are two of the most common one. One is called 5,3-PC, this phosphatidylcholine. And this 5 means there are 5 cyclobutane rings are fused, and 3-PC means there are 3 of them here. And these are the two common intermediate or the common building blocks of these molecules. Now, obviously, there has been a lot of interest in synthesizing these molecules. But the question is, why do you want to synthesize that? Because there are multiple reasons. This bacteria, anamoxosome, this happens, of course, in the absence of air and in dark often, and it divides once in only 2 to 3 weeks. Therefore, it is an extremely slow process. And along with this ladderin phospholipid, there are a lot of other lipids that comes with it. So therefore, it is almost impossible to separate into a very pure form of this molecule. So that makes it harder to do further studies. Therefore, laboratory synthesis remains one of the best methods for actually getting them in pure form and to study further biological function and possibly to answer some of the unanswered questions that I will come back afterwards. Now, if we look at this molecule, these are the 2 parts of this molecule. There has been a few synthesis, and mostly by these 3 groups. So the initial synthesis was done by Core's group, and then they have done an enhancer selective synthesis. But it is actually Noah Burns group in 2016, they actually made both the fragments, and they combined and characterized or established the absolute configuration of this molecule. So, and just to give you a very briefly how they made this molecule. So initially, they started with this racemic compound and did an enzymatic resolution of this molecule to prepare the building block. They went on and then they finally coupled this long chain. As I was telling you earlier, that people normally use an organometallic compound. This is an alkyl zinc compound that is coupled to make the final product. In Brown's strategy, they did an catalytic transfer of 2+2 cycloaddition at the early stage. Then this was taken to the final product over several steps. So basically, the common thing about both the synthesis is, of course, this is the same thing they're using, except the protecting group. But the stereochemical information was set at the very early stage of the synthesis. That means if you want to make the aride enantiomer, you have to start from the beginning and go through the whole process. We thought it can be done in a much simpler manner. Simply cut this bond. And now we can do it via symmetry breaking. We have these compounds, both of them, which are very symmetrical. And if you can stitch this here, we actually get this molecule. And this can be done at a very late stage of the synthesis. Of course, it is very easy to tell what we are planning to do. What we are talking about, coupling between two sp2ch bond. And there is no method known as of today. So therefore, this is an impossible transformation as of today. Even in the future, it's going to be challenging, because even if you are having sp3ch coupling, you can imagine in these two molecules the different types of CH bonds available. So pick one type chemoselectively, and then regioselectively, then stereoselectively. It is going to be a monumental challenge. So we thought, let's simplify that. Let's put some functional group. So I often compare this. This transformation would be almost if I ask you to climb this wall. There is no grip there. You will not be able to climb that. But the functional groups are basically grips. So now you go to a wall where you can do the rock climbing. There are some grips there. And once you reach there, you can remove those grips. So that's why you can actually remove the functional group, and then you can actually get to this step. That will be much simpler. So because in this case now we can break this, and this basically brought us to the reaction that I was showing earlier, where the functional group is nitro, and we can form the CC bond that I was talking about. So this is something that we can do. Now, there are two problems. One is, of course, on the drawing board that we are talking about whether we can do the enantiomer position selective conjugate addition, because this was our previous substrate, where this is the reactive site, which is somewhat closer to the substituents. And this is our present molecule, where this the other part of the molecule is actually very far from the reaction site, whether we will be able to get the desired level of selectivity. Second thing is, of course, how to make the symmetric molecule. So we first thought, let's solve the first problem. And we thought, let us start with some simplified model substrate. Instead of cyclobutane, we can have a cyclopropane, and that can be prepared very easily. We can do that. Now, when we optimized everything, my student Sion, actually, he was instrumental in taking up this project. And he could show that using this catalyst, now instead of urea or thiourea, we have something called squaramide. And using that, we can do the same reaction again using the nitroalkane. And then we actually prepared some cyclopropane-based natural product as well. I will not go to that. Now, having solved this problem, we went to this drawing board again, or on the bench rather, to find out how to make this molecule. Now, this was not unknown. We knew how to make the molecule. It was operationally difficult. And in general, if you look at ladder ends, one of the simplest strategy would be to cut this bond so that you can get a strand of polyolefins, and you can do a series of 2+2 cycloaddition reaction. Now, that is theoretically possible. It requires the parallel alignment of this polyene strand with a separation of 3 to 4 angstrom between the two strands. And because this is entropically highly unfavorable process, unless they are, of course, arranged already in a molecular architecture of this type, or it happens in a crystals. This has been known. It was reported already. But there are more systematic approach of preparing this type of ladder ends. For example, you can take these molecules I was discussing with Kishan today, that you have this cyclobutadiene, which is anti-aromatic. They're normally stabilized by this type of eta-4 complex with iron. And in the presence of oxidant, you can clip that, and then highly unstable cyclobutadiene immediately participates in a 4+2 cycloaddition 2 times to form this type of compound. They call pterodactyl adines because of their similarity with pterodactyl, that is the flying dinosaur. Now, this was acquired also by Professor Mehta's group with some functionalized cyclobutadiene, but we became interested in this report by Martin, where they have done actually systematically synthesis of this pterodactyladine. They have done a 2+2 cycloaddition with maleic anhydride and acetylene 2 times, and then they went on to make these molecules. So we thought this can be a good method for making our starting material. And this is the first reactions we had to do. And so this is basically the reaction setup we finally have with Sion set it up, the entire thing. And we of course thank Professor N.D. Pradeep Singh from IIT Kharagpur who helped us establish this setup. In fact, he allowed Sion to go to his lab, work on for 1 week to get some initial heat, and then he came back. Now, we need to make this molecule because we want to get this diol as an intermediate. It can be, of course, obtained from this diacid, which is commercially available only if your pocket is deep. Because this cost, 5 gram cost close to 1 lakh rupees. And we made this compound about 270 gram, which is essentially the same as this. You can calculate how much it cost. And of course, there is a trick. So you can see the reaction time. And this is the photochemical reaction. So this reaction was going on for 2 months in the lab because hours. And this, yes, and this was 2 months because we could not run the. Oh no, we have checked it. This was done, of course, in a very large scale. We have done it in a small scale, which works faster because we can't run the light for more than 6 hours. It heats up, so we have to cool it down and then start again. Overnight, we cannot run. So this was going on for 2 months, and then finally we got 270 gram of this final compound, this one. So which was great, and that cost approximately some 50 lakhs or something. Anyway, so that is besides the point. So we then finally converted this OH to the mesylate and then got this little compound, and then you oxidized, chlorinated, and then did the elimination. So this little devil basically took us 6 months to make because this is, again, all the steps are simple. Only thing is that this is highly volatile. It took us several attempts to find out the best condition where we of course can't purify that. We have to go with the impure one. But we can actually get this. Now, once we have that, we can actually do 2+2 cycloaddition. And so we can actually, now we have our starting material. So we are all set to do our alkylation reaction. So this is the long chain that we are trying to form by breaking the symmetry of this molecule. We can do that using a similar catalyst. And we got it in 69% yield, about 80% selectivity, and then we had to remove the functional group. We did by converting this indione to the corresponding diene, and then followed by hydrogenation. Now you can see that these functional groups are gone. We have this final compound. All we need to do is remove the TBS. We could do that, and this is 3-ladderenol. By simple functionalization, we got an extra structure that established the absolute configuration of the final compound. Now, the key features of our reaction is that we are introducing the stereocenter at a very late stage of the synthesis. So therefore, if we want to make both the enantiomers, we can do it very easily. We can also make the analogs very easily. We have made them. Now, having done 3-ladderenol, now you are wondering, can we make -5-ladderenoic acid? That is the more difficult part of that. But the question is, why do you want to make that? Because in natural product synthesis, of course, there are many fascinating looking molecules. One can go on and make them. But the question is, why do you want to make them? Is it good for anything else? Or is it going to be informative in some other aspect? So if we look at the common carbon skeleton, that is ladderenoic acid here, and I have put this dotted bond with different colors. And now so this is, of course, if they are all actual bonds, so this is the natural product. If we break the red bond, we get the other natural product, but there are 3 others bond that can form. So you can actually get all of them. So this is another natural product. So these 2 are part of the natural product. Now, these 3 compounds has never been isolated. Now, there is a possibility that they are also natural product. It has not been isolated yet. Another possibility is that now this was isolated from this membrane of this particular bacteria, right? Now, anamoxosome on this particular bacteria. Now, this bacteria has chosen this lipid for a particular purpose to avoid or to prevent the permeation of a proton across the membrane. Now, it is likely that nature made all of them, but then through the evolutionary process discarded the rest of them because they are not good enough. The only way we can answer this question is that if you can actually make all of them, and then you can test it, their proton permeability. So we thought of doing that. So for that, we need a unified strategy to make all of them. Now, this is something we have already made. So we actually need the unified strategy to make all 4 of them, including 5 dihyaluronic acid. If we look at closely, here the long chain is connected to the 4-membered ring. In the previous case, it was connected to the 6-membered ring. So therefore, we could easily apply our strategy for making this molecule. Now, this miso-indione, it is actually a requirement which basically makes this strategy useless for making the other 4 molecules. Unless, of course, we put another 6-membered ring here and then choose to delete that. Essentially, what I'm trying to do is, if we can break this, we can get to the starting materials. And once we have synthesized this one, we need to, so this is of course what we can do. And once we have that, we just need to delete this. Then the long chain is there. Now I'm going to show you how or what are the possibilities for deleting that. Now this is just a cartoon representation of that one for make it simpler. Now this is the possible what we call the deconstruction strategies. What we can do in this case, so this is of course the main reaction. We can delete these 2 carbon that I marked here, and that will lead us to this diketo. We can do the deoxygenation to get the compound. We can delete the 3 carbons here to get this keto, and then again we can do a deoxygenation to get this compound. Or we can delete a single carbon, and that will lead us to a different compound. The connectivity is now here. So therefore, we need to make the enantiomeric one using the different catalyst, but that is possible. Only thing what we need to do that, if we remove 2 or 3 or 1 carbon, depending on that, the chain length has to be modified. Again, that is very simple, can be done. Now, so this basically brought us to our synthesis, the strategy, where we have 3 different steps. One is this one to make the starting material. One is the enantioselective sp2ch alkylative desymmetrization. We know that works. That will work in this case. And then the deconstructive carbon deletion. We first thought of focusing on the carbon deletion approach using a model substrate, because that will be much simpler to make. This one we can make very easily, or even the long chain one we can make very easily. Now then, our first idea was we can cleave this double bond by ozonolysis to get an aldehyde or the alpha ketoaldehyde and one to diketone. And it is known in the literature that if we use this rhodium complex called Wilkinson catalyst, it actually removes carbonyl compound from alpha ketoaldehyde, aldehyde, or diketone to form this. So this 3 carbon deletion is we thought can be done, and you can get this ketone, and then we can of course deoxygenate that. When we actually attempted this reaction, instead of this ketoaldehyde, we got this one, which is actually the same as this molecule, just one water added here, that added, and then went back here. So it actually forms a complex acetal and or the ketal. We thought that these 2 can be in equilibrium, so we might be able to do the decarbonylation. So when we tried, it did not work. So on the other hand, we can do this ozonolysis in somewhat different condition. So we can delete already 1 carbon, and then we have a carboxylic acid and diketone. We tried the decarboxylation, but it did not work. We got something else. If we tried to convert this carboxylic acid to the acid chloride followed by decarbonylation, that also did not work. And these are only the few of them. There are many things we tried. Simon probably tried almost 6 months on this and nothing worked. So then we thought, okay, 3 carbon or 2 carbon deletion is not working. Let's go back to the 1 carbon deletion strategy. So now what we need is that we need to have a chemoselective deoxygenation if we want to delete this one. Once we have that, then we can cut this bond and we will get this, and then hopefully we will be able to delete this one by decarbonylation or decarboxylation reaction. So overall, this will be a deconstructive 1 carbon deletion approach. So let's focus on the chemoselective deoxygenation. We could do that by reducing this to ketone to the corresponding diol. And then by treating with a Lewis acid, we can actually eliminate one of them, the selective deoxygenation, so-called vinylogous pinacol rearrangement, because now BH3 can coordinate to this one selectively because this is sterically less hindered. And now this leads to a hydride migration and then followed by elimination of that. Only problem is that we have the double bond in the wrong position, but by using modified condition, we can actually bring it to the right position. So this is what we have now. So next, we need to do the CC bond scission. So for that, we first did the dehydrogenation, and then the ketone we converted to the alcohol. And then, of course, we remove the alcohol dehydration, we get this olefin, and now followed by ozonolitic cleavage of that. So therefore, the CC bond scission worked. Now the one carbon deletion is remaining. So with this one, decarbonylation did not work, but we can, with the carboxylic acid, we can actually do the decarboxylation through the corresponding or using a Barton decarboxylation under radical condition. It's a MOM, methoxymethyl protecting group. So sorry about that. I should have given the full name of that. So that's a protecting group actually. So once we have that, and then we can, so both of them we have done. Now only thing is that this has to be deoxygenated, that you can do under Wolff-Fishner condition. So this is basically the model. From the model substrate, you can go to the final product. So now this is just the whole summary what I showed here. Starting from this one, you can actually go to this for the model substrate. Total 11 steps, 11% overall only, but only 4 chromatographic purification. So now we could apply this. So this is what we could do now. This is already know that it works. So now basically this is what is remaining. And this is again, we have done using the same up to this point I've already mentioned. And this one, so we need to form now for getting these, we need to form 5 4-membered rings. And here, this is the first one, this is the second one I showed. This is the third one with 2 + 2 cycloaddition. Then we did a diazo here, followed by Wolff rearrangement and decarboxylation. So this is the 4th one. And after removing bromo, we get this double bond here. And so therefore we could actually get this. And now this one, if we do a 2 + 2, this is the 5th cyclobutane ring that we can generate in this case. And now we are ready with our starting material for the last step. So this is the step that is remaining now. So this is the step again. This is the long chain. What we did earlier, we can set it up using a very similar reaction condition. We can get the product with good enough yield and good selectivity. And now the end game is what I have showed earlier, that 1 carbon deletion approach. And then that led us to the final compound. And overall 6.4% yield, but only 4 chromatographic purification. So now, and this one has to be oxidized to get the ladderanic acid. And we compare the optical rotation with the one that is reported in the literature. We also match the NMR. This is the proton NMR. This is the literature one, and this is our sample, or the 13C NMR. It is matching perfectly. So therefore, we do have the -5 ladderanic acid. So now, as I showed that this was the one that was reported, we were happy to have our synthesis as a part of that. And there was a fun fact was in 2009, all 4 of us were working in the same building. So I was working with Professor Kure, Kevin was the postdoc with Professor Kure, Noah Burns was postdoc with Eric Jacobson, just 1 floor below us. So therefore, it was funny that in the whole world, we had the 4 groups worked on this, and 2009, all of us were in the same building. So now, coming back to that, we do have an unified strategy now. And as I mentioned, this is already what we have made. Earlier, we have made 3-ladderanol. We also made this inverse 3-ladderanol using the same strategy. Now, this is the truth that we are currently working on. Once we have all of them, we will be able to test the or try to answer the questions that I asked earlier. And so this is what I showed, that using our reaction or the desymmetrization reaction, we can make +3 ladderanol and -5 ladderanic acid and some of the other unnatural analogs of these molecules. So now, so this is what we began with our philosophy, and I was asking some of these questions like why to break symmetry, and I hope I was able to convince you that breaking symmetrical molecules, or the breaking the symmetry of a symmetrical molecules, could be a very attractive method for synthesizing some of the complex structures. So Sayan was the key student or the main person, almost single-handedly he executed the entire project. And only thing I told him when he joined my group that I want to use our reaction for preparing this natural products. The rest of the thing he did on his own. There has been a lot of frustrations, but he could overcome all of them. He could come up with new strategies every single time. And then he executed. Right now, he's writing a thesis. Subhajit helped him in one of the projects at the early phase of the thing. And now Sayan is training our new integrated PhD student, Prabha, So who will take on those two unnatural molecules that we are trying to make. Of course, the initial reactions were established by Madhu, Rahul, and Deepak also helped in some of the other projects as well. And this is my group from the last year. And I thank all the funding agencies for supporting our research over the years, and my collaborator Jennifer and Garima in some other projects, and former members. And I would like to thank you all for your attention. I'll be happy to answer any questions.
Santanu, this was fantastic. Such complex synthesis. So we'll have some questions and
These are the funniest lipids I have ever seen in my life.
Indeed.
So I'm not a synthetic chemist. I don't have a question about the synthetic part, but I was wondering about the packing. And you're talking about natural lipids
Right.
that occur in bacteria.
Right, yeah.
It's these are the tails are fat. So I'd expect the packing to be very hard.
Yes.
Or how does it prevent transport of protons and all that? It's very
Right. So
interesting.
there is a DFT study on the packing of these molecules. If you think that ladders are like this, right? So there are two types of packing this showed. So the DFT one is that so-called parallel packing, where and other one is on the side. So both cases, they say London dispersion force plays a major role. So they actually compared the packing of the ladderin molecules, or they did 3 ladderin, 4 ladderin, and 5 ladderin, and with the same chain length of the simple long chain aliphatic one. And they showed that the attractive dispersive interactions in the ladderin is much stronger compared to the long chain one, and in both the cases. So therefore, that is so dispersion possibly plays a major role in these reactions.
Yeah, I have a very general question. Why, I mean, you said in the beginning that making symmetric molecules is much easier than making antisymmetric. What is a general principle behind this? Because typically for single molecule, you have a symmetry, then there is always symmetry breaking because of Yang-Teller kind of effect, where
Right.
you have a one state going to minimum. So energy minimization actually will prefer such kind of a breaking.
Yes, you're right, because yeah, when you have the unsymmetrical one, so the chaos is more basically. So the entropy of the system will be much higher. Now, what I mean by breaking symmetrical molecule to go to an unsymmetrical molecule, it is not just to make the unsymmetrical molecule. It is just to achieve one of the two possible enantiomers. Basically, the handedness of the molecules, right? So one of them has to be obtained selectively. So if we do not break the symmetry, if we start from the scratch, at some point we have to establish the stereo center, and that has to be carried forward, like I showed in the previous examples. But once you have the symmetric molecule, like in this case, the entire skeleton is already established. And that time, just one operation will set the entire thing. There is nothing can go wrong. So that's what I meant by breaking symmetry. If it is a completely acyclic molecule, things can be difficult. Although there are examples known where even then, by breaking symmetry, one can actually achieve the unsymmetrical molecule.
okay, Shawna.
Very interesting emphasis. I was just wondering, the two things I want to ask. How do you ensure that it is going to remain in a symmetrical field out to the
Right.
The second is the definition of this reaction.
Mm-hmm. Right. Yes.
Have in the solution, again, it was solving the base.
Mm.
And do they form some superstructure continues?
Okay. Right. So yeah, just to answer, I think the first question was to sorry. Yeah. So in this case, so if we go to the, let me go to the synthetic scheme. Yeah. So in this case, so of course, up to this point, this is symmetrical. This is not chiral. But once we form this compound, so we are forming this CC bond, right? So now, if we if it has to racemize, this CC bond has to migrate from here to there. And that is not possible with any steps we are doing. In fact, that sort of a migration will be difficult in general, even if you want to do that. And all the steps, whatever we are doing, we are actually playing around this area. And of course, one key thing in this case, as I mentioned, that with our single carbon deletion approach, if we go to that. Yeah, so in this case, as I mentioned, that we have to do the chemoselective deoxygenation, either this one or this one. So that was the key. If we cannot control the chemoselectivity, then actually we will be in trouble. And luckily, in this case, we could do that because of the sterically less hindered nature of the other alcohol. Okay, so that is one thing. Other thing is that, yes, when you do the reaction, you characterize by NMR. So proton helps, 13C to some extent, but the most helpful is the DEPT. We can see how many CH2s are there, and that actually gives a very good amount of information. In some cases, we can crystallize them, and we can actually get the product. And when we have multiple, go to that. Yeah, when we have this type of molecules, like all 4-membered ring, we know generally these reactions, they do not fold. They are all so-called anti-cyclobutane fused system. So it's much simpler. And in solution phase, they are soluble in number of solvents. So we have never seen any aggregate in any of the solvent to form like a precipitate or any sort of crystal or anything. Of course, in some compounds we could crystallize, but normally we have not seen any superstructure. We have not studied the detail like concentration dependent aggregation and all so far, but normally we have not seen any aggregation.
Hello.
Yeah.
Yeah, very interesting talk. I somehow remember Professor Govardhan Mehta was talking about how to make bucky balls.
Right, right.
So it's very fascinating. So my question is that, I mean, you essentially selectively cutting some carbon-carbon bonds.
Right.
Why are the symmetry breaking playing a role? This is something.
That is the first step. Yeah, this is the first step, this step itself. This is the symmetric molecule. And now, so this is the CC bond we are forming in this initial reaction.
What I'm saying is that, is it that cutting that CC bond has any role to symmetry breaking? I mean, do you mean that it becomes more difficult if it involves breaking the symmetry, or typically, I mean,
no, breaking symmetry will make it easier. But if we have to build it step by step from the beginning, like instead of, I mean, of course, we can think of a strategy where we stitch it at the very beginning and the entire thing we are setting up next to each other. That could be a difficult strategy. But when we were preparing this one, we didn't have to worry about any selectivity because we knew all this 2+2, whatever is happening, that will happen in the all trans fashion.
Was where the symmetry is playing a role. I mean, that was something. I could see that, okay, you're following different steps, but
right.
Symmetry is playing somewhere. That was the thing which.
No, symmetry itself doesn't play a role except in the desymmetrization step. So otherwise, symmetry doesn't play much role. Yeah, symmetry breaking is actually the key step in our case. Yeah.
Less number of steps for symmetric molecule to achieve a symmetric molecule, but then that's what the idea is.
I was trying to understand is that whenever you are cutting a bond
right.
or something, does symmetry has any influence?
Okay, yeah.
I mean, that was the thing.
Yes.
I understand that the cutting is not trivial.
Yeah. No, but yeah, now I got it actually. So if you think of a symmetrical molecule and then if you cut a bond, then you do not need to work because there is only one product possible. But if the molecule is not symmetrical, then we cut a bond or form a bond, then there are multiple possibilities. So that's why the symmetry makes it, yes, simpler. Like I showed in the case of deoxygenation, like in this particular case, if it was here, I wouldn't have to worry which one is going. But in the other case, I have to worry because the CC bond is already formed there.
Just occur and then cut it.
Okay, yeah.
Okay. So what is known about the natural way in which this molecule is synthesized in the bacteria?
Yeah.
I mean, how does it make these squares?
Yes.
That is one.
Mhm.
And then my question is, if you can now feed any of your starting materials to something like an yeast and use a semi-synthetic pathway to
Make this molecule.
to make these molecules.
Yes. So, okay. So first of all, when it comes to the biosynthesis, in one word, it is right now unknown. As of now, it is unknown. There has been a few proposals. So this is one of them. So one of the proposal was that you have a completely saturated molecule, and there is a class of enzyme called desaturase, which can selectively introduce unsaturation in the molecule that can fold in something like that, and then it undergoes a radical mediated cyclization step by step. This is one proposal. So another thing, as I mentioned, that if we have two parallel strands of olefins, they can undergo series of photochemical 2+2 cycloaddition. But this enzyme resides at the bottom of the ocean most of the time when there is no light. So therefore, photochemical 2+2 cycloaddition is an improbable option. Now, this is something that was thought for a long time, this radical mediated cyclization. Now, there was a report where actually they have done the energy calculation for the reverse one. That means if you have a radical here, how it can systematically break to come to this molecule. Okay, so what they found out is the following. So this is, I took it from this paper. So this is this molecule from A. You can see that when you come from A to F, it is about 40 kcal exergonic pathway, right? Now, this reaction would be the reverse of that. And that will be 40 to 45 kcal uphill energy, so which will be extremely difficult. But one thing they figured out is that it is not a single enzyme that does this work. And they have identified, I think, a collection of 34 genes from this wastewater bacteria. And they, as a combination, they're actually doing this work. But again, there is no single proposition that is accepted as of now. There is one more, interestingly, where they showed that apparently there are enzymes known in the molecule in nature which does cyclopropionation. So another possibility, again, this is a theoretical study. They have done very loosely defined proposition that once we have this series of cyclopropane, they can undergo rearrangement to form this, and then desaturases come to the picture and then form this molecule. But again, these are all proposition. But yeah, it is unknown as of now. Yes. But what they did find out, like when I was talking about like which bond was cleaving, right? As I was trying to show, I was showing that all possible natural products like this one or unnatural compounds. So what is known is that this is somebody studied experimentally. If you take this molecule and they tried to see that there was a proposal that whatever we are isolating could be something very old enzyme that we are isolating now. So they tried to figure out how stable they are. So they systematically heated this molecule beyond 120. This bond and this bond, they cleave, both of them. So you get 6-4-6 scaffold. So this middle 4-member ring remains and the side one gets cleaved. So, and that compound is stable until 260 degree, and they could see by GC and all. Now, that's why, like when I'm talking about at least these two, so how they are formed and whatever biosynthetic proposition was made, they were for this 5-ladder in motif, not 3-ladder in. It is likely that it is formed by cleavage of one of these bonds. Now, if that happens, both are equally probable. So why only this one is forming, not the other one? So hence, this what we are thinking, that both of them can be natural product or not anymore. So that's what. But biosynthetic is unknown as of now.
So here are very basic questions. And if you do just first calculation, that will be energetically convertible or?
No, we don't do that. We I mean.
No, why I'm asking? Because you are starting from symmetry. The question is so that it will be energetically more right? Never do any reaction, final product. Whatever you go, stable, right? In a sense, minimum energy.
Right. So in our case, actually the important thing is when you have a symmetrical molecule, you have two sides which are identical, but anisotropy. Now, where the reaction happens, if we want to calculate, we have to calculate the energy of those two steps in the presence of our chiral catalyst, because those two transition states will be diastereomeric. So we need to have sufficient energy gap between those two transition states so that we can get one of them selectively. If they are energetically identical, we will never get any selectivity. So that is the main thing, our part, but we don't do that because we don't have expertise as of now. So if we require, we take the help of our collaborators. Yes, we have to do in every step, NMR, IR, mass, because that is of course to find out whether the compound is forming or not. And in a synthetic chemistry journals, it is also a requirement that every step has to be characterized. Only in some cases, like in our synthetic steps, as I mentioned, that let me go to that. Yeah, so what I mentioned that this is total 11 steps, but only 4 chromatographic purifications. So in some cases, we do not need to purify them. We can check the NMR to find out if it has happened or not, and then take it to the next step. Now, if it is too much of a mixture, we purify. Only when it is pure enough to be carried forward to the next step, we could do that. So in some cases, like we have, this is in industrial term, they call it telescoping, because A to B goes to C to D, and then in D we purify that. So we try to minimize the number of purification steps, so that because purification is economically not a good, more difficult. Yes. Yeah.
Thank you. That was Professor Karthik Ghosh. He has been a PhD alum of this institute. He is now in Missouri State. So, yeah, any more questions? If there are none, let's thank Santanu once again. And let's walk outside and have some snacks. Oh, actually, it's inside this time. Okay, so in the AC. Yeah, in the West Canteen. And thank you. I thank the auditorium staff for helping us. So.
No, we have done the integration.